Hello dear students, in today's class we will discuss the important features and the pathological characteristics of a protozoan parasite Endamoeba histolytica. Endamoeba is pathogenic and it causes intestinal amoebiasis or amoebic dysentery in humans, and this lives in the large intestine of man, but sometimes it is also known to cause extra-intestinal infections. That means in severe cases this will migrate through the blood vessels and enter organs like the liver, spleen, kidneys, brain, skin, etc., and they cause abscesses there. What do you mean by an abscess? It is a painful collection of pus in the body. So, such extra-intestinal infections are also known to be caused by Endamoeba histolytica, but its actual resident place is human intestine. And in the human intestine, the intestinal wall has got several layers. The innermost wall of the intestine is called as the mucosa, over which is the submucosa, above which is the muscularis, and above which is the serosa. And this Endamoeba, once it infects humans, it will be seen in the lumen of the intestine, the mucous membrane of the intestine, and even in the submucous layer of the large intestine of man. So, usually it is found in the human large intestine in its mucus, mucosa and submucosa, and also in the lumen. Different forms of the parasite can be seen in all these places. And as I earlier told you, along with intestine, some are also known to cause infections in other organs by migrating through blood. Like liver, you can see abscesses being formed in the liver. Regarding the distribution of Endamoeba, this pathogenic form, it is cosmopolitan in distribution. That is, it is seen almost worldwide, and it is frequently recovered from fresh water contaminated with human feces, with a higher prevalence in countries of low socioeconomic status and poor public health, because when a person is infected with Endamoeba, his feces will carry cysts of Endamoeba. So, these cysts are infective forms. So, through the infected person's feces, the cysts will come out, and if this feces contaminates water or other or anything which is consumed by man, what happens is that it enters into human body. So, in those places where poor sanitary conditions prevail, there is higher occurrence of this Endamoeba infection, and 90% of Endamoeba histolytica infections are asymptomatic, that means without symptoms, but 10% are symptomatic, and nearly 50 million people become symptomatic, with about 1 lakh deaths yearly. Then coming to the host of Endamoeba histolytica, its predominant host is man. Man is the definitive host, and also primates. And there are also cases where mammals such as dogs, cats, etc. become infected transiently, but they are not thought to contribute significantly to transmission. But in China, it has been found that dogs serve as possible sources of human infection. Now, coming to the stages in Endamoeba, in the life cycle of Endamoeba, you can see Endamoeba in 3 morphologically distinct forms. They are, this is called as trophozoite, this is called as cystic form, and in between there is a pre-cystic form. So, these are the 3 forms in which you can see Endamoeba histolytica, that is the trophozoite, the pre-cystic form, and finally the cystic form. Now, the trophozoite form, it again occurs in 2 stages. It can again be divided into 2 stages. This is called as the magna form, and this is called as the minuta form. Now, where are all these forms found? The pre-cystic form, the cystic form, and the minuta form of the trophozoite are found in intestinal lumen. But this trophozoite form, this magna form of the trophozoite, it is seen in the mucous and submucosal layers of the wall of large intestine. So, the magna form of trophozoite resides in the mucosa and the submucosa, whereas the minuta form of the trophozoite the pre-cystic form and the cystic form, they are seen in the intestinal lumen. Now what is this trophozoid? Tropho means feeding. So trophozoid is the fully mature feeding and growing vegetative form of the parasite. That means it is not the sexual phase, it is not the reproductive phase, but it is the vegetative phase which is involved in feeding. And as I already told you, there are two forms, that is the magna and the minuta. So from the name itself, magna is the larger form. It is more harmful. It feeds on human blood cells. It feeds on the intestinal mucosal tissues, and it is also able to cause ulcers in the wall of the intestine. The minuta form is smaller. It is a harmless form comparatively, and it lives in the intestinal lumen. So we will go to the trophozoid. The trophozoid is larger, it is irregularly shaped, and its cell is covered by a plasma membrane, and the cytoplasm enclosed inside the plasma membrane can be divided into two regions, that is an outer clear glassy hyaline ectoplasm, as you can see here, and an inner granular endoplasm. So this is the granular endoplasm. Now the endoplasm contains a single nucleus, as you can see here, and there are many food vacuoles. It consists of many food vacuoles, and the cytoplasm is granular. Mitochondria are absent. Endomembrane system is also very simple. And what do these food vacuoles contain? As it was mentioned, this magna form is found in the mucosal and submucosal layer of the host intestine. So it is feeding there, and these food vacuoles, they will be containing the ingested human RBCs, the tissue debris, the tissue waste, bacteria and digestive enzymes. So all these will be there inside these food vacuoles. Whatever it has acquired from the human intestinal wall will be there inside these food vacuoles. And the presence of human RBCs in the food vacuoles is a diagnostic feature of Entamoeba histolytica, whether it is the infective form. And the presence of RBCs inside the food vacuoles indicates that the parasite feeds on human tissues. Now coming to the pre-cystic form and the cystic form and even the minuta form, there are no food vacuoles. Food vacuoles are absent. Now coming to the nucleus again, it has got a spherical nucleus which is covered by the nuclear membrane, and the genetic material is distributed in the form of chromatin. Chromatin is distributed in two forms. There is a central region which is called as the karyosome. So this is a central mass of chromatin which is called as the karyosome, and attached to the inner lining of the nuclear membrane, inner region of the nuclear membrane, there is granular mass of chromatin, and it is called as granular or beaded chromatin. So in short, the genetic material or the chromatin is distributed in two forms. There is a central mass of chromatin, and there is a peripheral granular layer of chromatin attached to the inner surface of the nuclear membrane, and in between there is a clear, in between the central mass and the outer granular region, there is a here there is an region which is devoid of any chromatin. So it's a clear achromatic or a chromatin free zone, and it consists of a number of this chromatin free zone consists of a number of fine radiating fibrils which is called as the linin network. So this is regarding the cytological features of Entamoeba histolytica, and you can see there is only a single pseudopodium. This is a parasite which possesses pseudopodium, and the single pseudopodium, it is flat, blunt and lobular, and so it is called as the lobopodium. It can withdraw the pseudopodium from one direction and extend it to a new direction whenever required. Now we come to the pre-cystic form. I told you pre-cystic form is an intermediate stage. This is the pre-cystic form. The pre-cystic form is an intermediate stage between the cystic and the trophozoid forms. So it is very small, it is rounded or ovoid in shape and has got a large nucleus, blunt pseudopodium projecting to one side. This is the pre-cystic form, projecting to one side, and food vacuoles, ingested RBCs, etc. are absent. So this is the precystic form. This is precystic form, sorry. Then coming to the cystic form. So this is the final cyst. So this is the encysted stage. That means it has got a protective covering. So this is the encysted form. So in early cystic forms, the endoplasm contains a mass of glycogen. It will have glycogen and some rods. You can see some refractile rods which are called as chromatoid bodies. These chromatoid bodies are actually considered to be crystalline aggregations of ribosomes. And you can see a glycogen vacuole where glycogen is stored. Now, as the cyst matures, so what happens as the cyst matures? You can see the glycogen reserves disappear, the chromatoid bodies disappear, and you can see the division of the nucleus. Nucleus divides 2 times to form a tetranucleate stage. So this is the beginning, then it acquires a protective covering, then it will have a glycogen reserve and a glycogen vacuole, chromatoid bodies which are aggregations of ribosomes, and gradually it will be losing this glycogen vacuole and the chromatoid bodies, and the nucleus divides twice to form a tetranucleate cyst. Now we move on to the life cycle of Endomoeba. It is a monogenetic parasite. That means the life cycle is completed in only one host, that is man. There is no intermediate or carrier host. And the life cycle is completed in 5 main stages, that is the minuta stage, the magna stage which was mentioned earlier, pre-cystic stage and the cystic stage. The cystic stage can itself be divided into 2, the encysted stage and the metacystic stage. So we will just have an outline of the life cycle. It can be divided into stage 1, stage 2 and stage 3. So stage 1 is the infective stage, where infection occurs by ingestion of mature cysts through fecally contaminated water or food. That means if in the feces of an infected person, there will be the cysts of this parasite, and if this feces contaminates water or food, and if it is consumed by another human being, he gets infected. And this cyst, as it was already mentioned, is having a protective wall, so it can survive several days and weeks in the ground or in water, and they are responsible for transmission. So that is the infective stage. Now the parasite has entered human body. Inside the human body, the stage 2 will be carried out, that is, it is called as excistation. That is, it is the process by which the cysts transform to trophozoites. And when the cysts enter the ileum of the small intestine of the host, the process of excistation begins. That means it is coming out of the cyst wall and it is forming a trophozoite. Trophozoites are released in the small intestine, and from here they migrate to the large intestine, because large intestine is the actual dwelling place of this parasite. So that is the second stage, which can be called as excistation. That means how the cyst develops into a trophozoite. And the stage 3. In stage 3 is the damaging phase. That is, the trophozoites, they multiply in the small intestine by binary fission to produce cysts that will exit through the human stool. And many trophozoites will remain inside the lumen of the intestine, and the others, what do? So these trophozoites, some of them develop again into cysts, some of them remain in the lumen of the intestine, and what the others do? They will attach themselves to the intestinal mucosa. They can even feed the tissues there, and they can even enter the bloodstream and further grow in the extra-intestinal regions of the host, like the lung, liver, brain, kidney, spleen, skin, etc. So this is the life cycle of Endomoeba in a nutshell. Now we will go to the details. So we will start with the infective phase. This stage, which is called as the tetranucleate stage, there are 4 nuclei. This stage, called as the tetranucleate stage, is the infective stage of the parasite. It passes to the outside along with human feces, and it can remain alive in the soil for some time under favorable conditions of temperature, moisture, etc. It is resistant to environmental conditions, but it is destroyable by heat, drought, etc. And it has been studied that it survives in water up to 8 months. It can survive in moist feces up to 1 month and in moist soil up to 10 days. During this period, it does not undergo any further development. Its further development is possible only in a human host. And so the transmission is contaminative. That means when the human being he consumes contaminated food or water, in fact having these cysts, he will get infected. So it is contaminative transmission. And sometimes the house flies and other insects also play some role in contaminating food and water with the cysts, because a house fly which will sit on feces, if it comes to uncovered food, again there is chance of infection. Now, fecal contamination of drinking water, food and vegetables also contribute much to the transmission of the infective cyst. Now, so as we mentioned, the cyst is ingested by the host. It is the tetranucleate stage which is ingested by the host. So he swallows it. The cyst reaches his small intestine, and it is absolutely not infected, it is not affected by the action of human gastric secretions, because there are powerful digestive enzymes produced by the human body in the stomach. There are protein digesting enzymes, but the cyst will survive all these because it has got a protective wall, and it comes to the small intestine. The last part of the small intestine, that is the ileum, or the beginning of the large intestine, the cecum. What happens is that the cyst wall will get ruptured. The wall of the cyst will get ruptured and it gets dissolved, and the parasite is now set free. The quadrinucleate, the tetranucleate parasite is now set free. That means it is coming out of the cyst wall. This is called as excystement or excystation. And this excysted tetranucleate endamoeba, it is called as the metacystic stage. So this is called as the metacystic stage. So how does excystation occur? How can it come out of the cyst wall? During excystation, what happens is that there will be the protective covering called as the cyst wall, the cytoplasm. The cytoplasm will slowly start retracting from the cyst wall, and it gets detached from the cyst wall by vigorous amoeboid movements. So it is there inside the wall. Now the cytoplasm slowly start retracting, and it will somehow get detached from the cyst wall by vigorous amoeboid movements. Simultaneously, the intestinal secretions dissolve some part of the cyst wall. So when these two processes combine, there will be breakage of the cyst wall. Through the split thus formed, the quadrinucleate amoeba comes out. So this is called as the metacystic form, and this process is called as excystation. Now what happens to this metacystic amoeba? The metacystic amoeba undergoes nuclear division, and there are 4 nuclei. When these nuclei undergo division, there will be 8 nucleus. So now inside this endamoeba, that means the metacystic form, there will be 8 nuclei. Now what happens is that the cytoplasm slowly gathers around each nucleus, and what happens? This will divide. There will be 8 nuclei. Around each nucleus, cytoplasm will gather, and finally it will divide to form 8 uninucleate amoebulae. So these are the uninucleate amoebulae. Now they will grow to mature metacystic trophozoites in the colon. Colon is a part of large intestine. So in the colon, they will start growing. They will grow to mature metacystic trophozoites. Now, what do these trophozoites? So these are the trophozoites which will, after growth, they become trophozoites. These trophozoites again undergo repeated binary fission and they give rise to large number of minuta forms. They will give rise to large number of minuta forms. And these minuta forms, they will invade the intestinal wall and grow to magna forms. So, the trophozoites divide into a number of minuta forms, and some of the minuta forms will enter into the intestinal mucosa and submucosa and start feeding on the tissue there, while some of the trophozoites undergo encystation and develop into new cysts. Now we have to see what happens with the trophozoites. So we will just compare the magna form and the minuta form. Minuta form is smaller than magna form. It is non-pathogenic. It lives in the lumen. Sometimes it becomes virulent, invades the intestinal wall and becomes the magna form. Magna form is larger. It is harmful. It is seen in the intestinal wall. It destroys the intestinal tissues and causes intestinal amoebiasis. Now, again, in another region we will see what happens. After some time, the magna forms, they will come out to the intestinal cavity. They lose their food vacuoles. I told you in the magna form there are food vacuoles which contain RBCs, human tissue debris, etc. Now, some of these magna forms, they will come out to the intestinal lumen. They lose their food vacuoles. They become rounded or ovoid and transform into a precystic form. So, here you can see some of the trophozoites will become precystic forms. The precystic form synthesizes glycogen, stores glycogen, it synthesizes chromatoid bodies, becomes spherical, develop a protective wall and develops into a cystic form. So, this is a uninucleate stage. It is an immature cyst which is formed from the magna form of the trophozoite. So, magna form of the trophozoite has now got encysted into a mononucleate cystic form, a precystic form you can call it. So, the wall is double walled, it is highly resistant, it is protective. It is mononucleate in condition. As it matures, its glycogen reserve and the chromatoid bodies gradually disappear and its nucleus undergoes two successive mitotic divisions. The first division will result in a binucleate cyst and the second one in a quadrinucleate cyst. So, this is the quadrinucleate cyst. This will come out through the human feces. So, this is the mature cyst which is quadrinucleate and it is that form which comes out of the human feces, and the life cycle is repeated again. So, we will just have a look again, that is the quadrinucleate cyst is ingested by man. It comes to the small intestine of man where it undergoes excistation. Then it divides into 8 uninucleate amoebulae. They will divide by they will grow and develop into trophozoites. These trophozoites undergo binary fissions. Some will develop into minuta forms which will stay in the lumen. Others will develop into magna forms, that is the minuta forms will develop into magna forms which will invade the intestinal wall, and some of these magna forms will come out, undergo encystation, first in a mononucleate stage, then in a binucleate stage, and then transform to the quadrinucleate stage which comes out through the human feces. So, these are the various stages in the life cycle of Entamoeba histolytica. The 10% show of the symptoms. And I told you it invades the intestinal wall, it multiplies there in large numbers and causes the destruction of intestinal tissues. The death of intestinal tissues will result, and for this it is having a tissue dissolving enzyme which is called as histolysin. So, this eventually leads to intestinal amoebiasis. So, the person suffers from inflammation, lesions, that means wounds, and ulceration of the large intestinal wall. The cecum as well as the ascending colon of the large intestine are the most commonly affected areas. and there will be discharge of blood, pus, etc. to the intestinal lumen, and they will come out along with the loose feces, and this is called amoebic dysentery. So, it is characterized by loose feces which is having mucus, blood clots, pus, etc. The incubation period of this endamoeba, it can range from days to 3 months, and the average period is 20 to 30 days. The disease onset is usually gradual, and the clinical symptoms include poor appetite, that means loss of requirement, appetite is lost. So, poor appetite, then inflated abdomen, there will be severe headache, gripping abdominal pain, frequent discharge of fluid feces, diarrhea, presence of blood and pus in the stools, etc. Frequent passage of stools is often attended by cramping. It is associated with cramping, that means pain in the abdomen, and tenesmus. What is meant by tenesmus? Even if the person he passes feces, even if he defecates, he will still have a feeling to defecate again. That is what is called as tenesmus. Even after defecation, he feels to defecate again. And the severe symptoms, they usually last for 4 to 6 weeks. Then slowly the abdominal pain gets reduced, the stools become normal, and usually the condition improves if treated. If the disease is left untreated, there can again be infections which can be called as relapses, and the disease becomes chronic. It can persist for years. And chronic amoebiasis is characterized by still severe symptoms, that is anemia. What do you mean by anemia? The reduction in RBC count. There will be reduction in the hemoglobin count of the blood. Cachexia. Cachexia means muscle loss or weight loss. Amoebic granuloma, that means tumor like growth in the large intestine. Multiple ulcers in the large intestine, and occasional passage of blood and mucus in the stools, inflammation of the large intestine, etc. And in acute amoebiasis, you can see deep ulcers formed in the intestinal wall. There will be perforation of the intestinal wall, hemorrhage or bleeding in the intestine, extreme constriction of the intestinal lumen, etc. There are other complications also which can be called as metastatic or invasive amoebiasis, as already mentioned, where the trophozoites, they can breach the intestinal walls. They will enter the bloodstream and travel to internal organs like liver, spleen, brain, kidney, testis, etc. So, these are the various complications arising out of Endamoeba histolytica infection, etc. So, these are some of the treatment regimes. So, this finishes the pathophysiology and the characteristic features of Endamoeba histolytica. Thank you.